Okay, what about the third messenger? Here we have a figure that demonstrates the third messenger protein kinase. So we have this process of activating a third messenger kinase through cyclic AMP. So you can see in this figure, this illustration, by this. No, that's not good. Anyways, okay. So this figure illustrates activation of a third messenger protein kinase through the second messenger cyclic AMP. So neurotransmitters here begin the process of activating genes by producing a second messenger cyclic AMP here. So and as we said before in the previous video about about how a first messenger can produce can lead to the second messenger. We've talked about that in previous video. So some second messengers activate intracellular enzymes known as protein kinase, like in this example. So we have inactive protein kinase here. So this enzyme, the protein kinase, is shown here as inactive, but with something called R here, which is the regulatory unit. What's regulatory unit? Regulatory units actually they regulate the enzyme. That's how they inactivate the enzyme. So okay, so that's it. So that would dissociating them from the protein kinase dimer. The second messenger would dissociate the regulatory units to activate the protein kinase, as we can see here. So we have second messenger that second messenger dissociate the regulatory regulatory units, and when they are dissociated, so the dimer would be active as separate proteins, as you can see here. So that's it. That's about the third messenger kinase. Cyclic AMP. Okay, cyclic AMP, that would dissociate the regulatory units from the dimer here, and then the third messenger active protein kinase would be ready to work and to phosphorylate things. Okay, so that's it. Let's just take a look about on phosphatase, and then we will get back to some kinase things. But let's just take a look on the other hand of this process of third messenger activation. Okay, here we have this figure that demonstrates the third messenger phosphatase and the activation of the third messenger phosphatase, and that would be through calcium, as we said before. So here we have a second messenger as calcium binding to an inactive phosphatase known as, for example, calcineurin here. This is inactive. When calcium is bind to that, this would activate the third messenger enzyme, and that would, so when it is active, it would work as a phosphatase. So that's it work. It would remove phosphate from fourth messenger phosphoproteins and so on. So let's get back to the kinase work. So third messenger kinase is activated now. What it can do, it actually put phosphate on critical proteins. Like what? Like these kinds of proteins. Let's just take a look at this. This is important actually. So here as we said, first messenger that leads to second messenger that would, work with the regulatory unit to activate the third messenger. So here we have a third messenger that is activated. We are talking about kinase messenger here. Kinase. We have two types, kinase and phosphatase. So here the activation of a third messenger kinase that would lead to add phosphates to a variety of phosphoproteins, such as this is like ligand-gated ion channels, voltage-gated ion channels, and various regulatory enzymes. So adding a phosphate group to some phosphoproteins activates them, but for other proteins this inactivates them. So that would not mean it activates all of these things. It adds phosphate, and according to that protein, the ligand-gated, voltage-gated, or the regulatory enzyme, according to that particular enzyme or protein, things would work. So, but that's the general principle of that. What about phosphatase? It's the other way around. So this is how third messenger phosphatase undo what kinase create. So it takes phosphate of critical proteins. So it is the opposite. So in contrast to the previous figure, the third messenger here, which is phosphatase, this enzyme removes phosphate groups from phosphoproteins, such as ligand-gated ion channels, voltage-gated ion channels, or regulatory enzymes. And again, again, actually, removing a phosphate group from some phosphoproteins would activate them, but removing phosphate enzymes from other enzymes or proteins would inactivate them. So it's not a thing. It's not by itself. It's not defining anything to remove or to put a phosphate group. So according to that protein or enzyme, things would be activated or inactivated. But in general, phosphatase would break down phosphate, kinase would add phosphate. As simple as that.